seminar we have planned after that will be October 23rd, which will be sort of implementing the tips that we learned from the October 9th session on networking and really getting to know our fellow trainees in the Metab trainee community. And also as part of this initiative, starting next week after the Metab talks, we'll have open networking. So if everyone can stick around, that would be great. And we can just discuss whatever is going on in our lives at that time, or discuss the talk that was just given. So follow us at Metab Trainees for more information, and that's on Twitter. And thank you, and I hope to see everyone there.
Okay, thank you, Adil. So I hope every one of the trainees will also follow the Twitter account and also log into the trainee sessions. So I think this is a great initiative, and congratulations on this initiative. But today we're going to continue with Sabine Steffens, and Sabine is a professor at the LMU, at the Institute for Cardiovascular Prevention. And Sabine has spent most of her scientific life studying endocannabinoids in cardiovascular disease. And I think today she will give a great overview on the role of the endocannabinoid system in atherosclerosis, and I think also in myocardial infarction and heart failure. Well, Sabine, please share your screen, and we will be listening. Sabine, you're still on mute, I think.
Okay, so yeah, today it's my turn to present what my lab is interested in. And yeah, the main really key topic we are interested in is the endocannabinoid signaling. And I would like to explain why we think this is really important in vascular inflammation and cardiac disease. So we are mainly interested in vascular inflammation and the process of healing and remodeling after an acute myocardial infarction. So the idea, the aim of our research is to identify new targets, and we are focusing on these endocannabinoid receptors to potentially limit this inflammation, prevent an acute event, or even to shape the repair process to a better healing response to hopefully prevent heart failure in MI patients. So what are endocannabinoids? They are lipid mediators. We call them bioactive lipid mediators. What does it mean? They can sense the environment of a cell or a tissue to react accordingly, to induce an intracellular signaling response. And this may lead to cell decisions such as proliferation or differentiation or apoptosis, so really generally speaking. So where do they come from? They are produced by membrane phospholipid remodeling in specific pathways. The main pathway for one of the endocannabinoids is maple phospholipase D, and the other main endocannabinoid, 2-AG, which is 2-arachidonylglycerol, is produced by deacylglycerol lipase. So in these ligands, they bind to main cannabinoid receptors, the CB1 receptor and CB2 receptor. They have been identified originally as the main receptors that mediate the psychotropic effects of cannabinoids. So especially the THC, it can bind to the CB1 receptor, and this is responsible for these psychotropic effects of marijuana. And there's also the CB2 receptor, which can also be activated by THC. We are now more focusing on the endogenous ligands, which bind to these receptors. As I said, they are rapidly metabolized from the membrane phospholipids, but they are also rapidly degraded by specific pathways, mainly the fatty acid amide hydrolase pathway, and the 2-AG is metabolized by monoacylglycerol lipase, and this then leads to arachidonic acids. So endocannabinoids can either act by binding to the receptors directly, or potentially indirectly. Endocannabinoid increase could lead to inflammatory effects by downstream mediators such as prostaglandins and leukotrienes. So I would just like to highlight this is there's an increasing network which is called endocannabidome. So we are so today focusing mainly on the key classical cannabinoid receptors here in this talk, which is the CB1 and CB2 here encircled, and there are also the main endogenous ligands. But there are also other receptors, G protein-coupled receptors, which are can be activated by these endogenous ligands. And there are also endocannabinoid-related mediators, which could also bind, for example, to cannabin receptor CB1 or CB2. So we are also in other projects studying the role of GPR55, for example, and GPR18. But today, as I said, it will be mainly about CB1 and CB2 signaling. And yeah, this is just to clarify here this overview that we have these classical lipid families where the endocannabinoids belong to, but we also have lipoamino acids here that could, for example, bind to the GPR18 receptor. And there are also still neurotransmitters, for example, serotonin, which could also bind to some of these receptors. So the best understood function of these endocannabinoids is certainly the function in the central nervous system. So there is the modulation of synaptic plasticity. When there is, so what does it mean? So there is an activation of a postsynaptic neuron, and the calcium then leads to endocannabinoid production. Then this leads to the presynaptic neuron activation of the cannabinoid receptor. And then this leads to inhibition of neurotransmitter release. So this is important, for example, in situations where there's a stress response, pain, and so on. So endocannabinoids may dampen this signaling, the neural transmission. And this is also important, for example, for appetite regulation, memory, and so on. And that's why this endocannabinoid system is considered as an important therapeutic target in neurological disorders. But so there's also not the central regulation. There are also important functions basically all over the body, and especially also in metabolism. So probably interesting for people here working. And then there is also the role in the cardiovascular system. For example, the regulation of vasodilation and cardiac function. Here. We are mostly interested in the inflammatory effects, and this is known for a long time that endocannabinoids are involved in immunomodulatory effects. So there is for example, if there's an infection or tissue injury, there's an upregulation of endocannabinoid levels, and this contributes also to immune cell recruitment. And as this immunomodulatory effect is thought to be mainly regulated by CB2 signaling, this is what we've studied mostly so far, and together with others. So when we started to work on this endocannabinoid system, we really started with this plant cannabinoid, THC, to actually see and confirm that this has a strong anti-inflammatory effect, and this also leads to reduced plaque formation in atherosclerosis mouse models. Then later on, we moved to more specific CB2 agonists, synthetic agonists that also show that CB2 overall seems to play an atheroprotective role. And there are other groups that also confirm this by knockout mice and hematopoietic bone marrow chimeras, for example. So more recently, then we went also to be focused on this downstream pathways, the MAC lipase pathway, which is important for the breakdown of 2-arachidonoglycerol, to see what are actually the endocannabinoids doing in this atherosclerosis disease process. So we started to look into this with a knockout mouse model where we have a global deficiency of MAC lipase. And this told us in an early time point when we have these mice on ApoE background, we give them a Western diet, they have less plaques in developing, and this in this early stage, and we could also confirm this with the pharmacological inhibitor. So now the question is, of course, which receptor is mediating this effect? What happens if we chronically block this degradation pathway? We have a systemic elevation of the 2-AG levels. And then there is a phenomenon which has been reported for the CB1 receptor. So if you have an activation of this G protein coupled receptor, there is an internalization. There is a chronic stimulation of this receptor. You can have a phosphorylation and then a beta arrestin mediated so-called desensitization. So in other conditions, in neurological models, it has been found if you have a inhibition of MAC lipase enzyme, there is over time a desensitization of the CB1 mediated response, for example, in whatever neurological readout. So we may speculate then when we have a pharmacological inhibition, maybe the CB1 receptor gets desensitized. And therefore we looked first of all into the protein levels, and to do so we used brain samples of mice treated with this enzyme inhibitor because we can there nicely detect the CB1 receptor. It is highly expressed, and there is also no CB2 cross-reactivity. So what you can nicely appreciate here, when the mice were exposed to this inhibitor over time, after 2 days, 3 days, they're really losing the CB1 signal, so the receptor is just degraded. Whereas this is the so-called, what we would describe as desensitization. So whereas there is no effect on the CB2 receptor. And the CB2 receptor was detected in the spleen because this is the main receptor on immune cells. So now we could argue that either this is due to the desensitization, so that there is no CB1 signaling anymore, or that the CB2 is activated here in this case of MAC lipase inhibition. To then further address this, we used also CB2 knockout mice, again on ApoE background, again fed the mice on high-fat diet and treated this MAC lipase inhibitor. And here this response, this atheroprotective response was then gone in, that would tell us that actually the MAC lipase pathway inhibition is CB2 dependent. So what immune cell type is then possibly mainly involved in this response. We, according to the literature, this is really historical reports that B cells express highest level of CB2 receptors among all leukocyte subsets. But this is also confirmed when we look now, for example, into the ImGen database, that among all immune cell subsets in the mouse, the B cells express highest CB2 levels. So, and there's also some information available about the role of CB2 in B cells. It plays an important role in the maturation in the bone marrow. And it's also important for the marginal zone B cell repositioning. So CB2 receptor is really important for shaping the B cell immune cell repertoire. And we know in the atherosclerosis context that B cells have different subsets. There are more pro-inflammatory, pro-atherogenic, and the anti-atherogenic B cell subsets, in particular the B1A subset. And these cells, the B1A cells, are known to produce natural IgM antibodies, which can bind to oxidized LDL, and this leads to foam cell or inhibition of foam cell formation. And it also helps to clear apoptotic cells, as these IgM antibodies also recognize epitopes on apoptotic cells. So when we looked into the B cell frequency in our mouse model, in this case here, this is the MAC lipase knockout model here, we see an increased frequency of the B1A subset, the atheroprotective phenotype, in case of MAG lipase deficiency. So suggesting that if we have an enhanced signaling via the 2-AG endocannabinoid, we have a preferential stimulation of these B1A subsets by CB2 signaling. And this is also in line with the increase of the IgM levels we saw in the mice in the plasma, and not only general IgM, but we also looked for the oxidation-specific epitopes. So to put this here on the summary cartoon here, we have in case of MAG lipase pathway targeting, inhibition, or knockout, we have elevated 2-AG endocannabinoid levels, which then can mediate an atheroprotective B1 response with an increase of IgM. So far, however, we don't know so much about the CB1 receptor. On the other hand, it is suggested it plays an opposite phenotype, a pro-atherogenic phenotype, which is in part regulated or linked to metabolic effects, as it plays an important role in the metabolism, and less known is about potentially immunomodulatory effects of the receptor. So as to this, the metabolic properties, this is a bit a busy figure. It's just to illustrate actually that it's doing a lot, the CB1 signaling, where the most or the first discovered was certainly in this hypothalamic, the hypothalamic CB1 signaling, which is important for the regulation of food intake. And CB1 knockout mice, conversely, are protected against obesity. But there is not only this central effect, there's also a lot of peripheral CB1 signaling in, for example, in adipose tissue, in the liver, and all of this may contribute to this important role in metabolic disorders, such as diabetes and obesity. So this important implication of the CB1 receptor in obesity then had led to this clinical studies here from with the cannabinoid receptor antagonists, rimonabant. You may be aware of this. It's almost 15 years ago. Yeah, that's more than 15 years ago, where this was studied with rimonabant in the so-called rimonabant in obesity trials. And they looked into the effects, the potential benefit on weight reduction, and also an overall improvement on cardiovascular risk factors. So, for example, in this study here, in this first study that was published from this consortium, shows you that there's a nice dose-dependent reduction of the weight, so less weight gain, I would say. In dose dependently with the rimonabant, so the CB1 antagonist. And there's also a nice improvement of HDL cholesterol. So this is increasing dose dependently, and triglycerides were downregulated. And there are also other parameters that were also positively affected. Unfortunately, the rimonabant was associated with severe psychotropic side effects, depression, and so on. So these clinical studies later on were stopped, and also rimonabant, that was already approved as an anti-obesity drug in Europe, was then again withdrawn from the market. So somewhat disappointing, a bit disappointment in the cannabinoid research community, but that's not the end of the story. So people went on with studying the peripherally selective CB1 antagonists. But I would just like to also point out that nevertheless, this CB1 antagonist was also tested in atherosclerosis models, in LDL receptor knockout mice. Mice were treated with this compound, and then, yeah, not so surprisingly, of course, when you treat mice with this CB1 antagonist, there's a clear effect on the weight, so the mice gain less weight. So then the question is, of course, if you see atheroprotection, is this weight dependent or independent effect? So then they also did include in this study pair feeding experiments as an additional control, meaning that mice had restricted food that they received to actually mimic this reduced weight gain that you see in the rimonabant group. And then, yeah, the question is what happens with the atherosclerosis. And I should also point out that the dose they used here was rather high compared to the clinical dose here, was 50 milligram compared to this 5 or 20 milligram in the clinical trial. So yes, rimonabant leads to reduced plaque formation. Here they claim there's no effect on this reduced or on the pair feeding. So if mice have access or have reduced access to food, this, according to this data, does not affect the plaque size. But still, there were quite some changes also in lipids. And as to potential inflammatory effects, this is less clear here from the study, what CB1 might do in atherosclerosis. So as I already pointed out, so this strategy with these first generation of CB1 antagonists, such as the rimonabant, is certainly not the way to go, because there's just this problem with the central nervous system effects of this global targeting. So then studies went on to develop drugs which would not pass the brain barrier. So these would selectively act on peripheral CB1 receptors, and this there are already quite some data out that show that this also has a beneficial effect. So this might be also a promising strategy for diabetes and obesity treatment potentially. So as we are more interested in atherosclerosis, inflammation, we look rather than looking on the metabolic changes on lipids, we are rather targeting more selectively the effects on immune cells or on vascular cells that might be affected by CB1 signaling, having in mind that whether this may be eventually be a potential target for treatment with a peripherally restricted CB1 antagonist. So we would like to really understand what it's doing in different compartments. I would just like to give you a short, yeah, maybe as a teaser, why we think this might also play a role in the bone marrow is this observation. So this work is led by a postdoc in the lab, Raquel, together with George, who is a PhD student. So they have, this is based on the observation that in CB1 knockout mice, we have less hematopoietic stem cells, as for example here illustrated in this colony assay. This is also linked to reduction of CXCL12 and stem cell factor, which is important in this regulation of the bone marrow niche. So, and we are also interested more specifically in cellular or cell-specific effects using, for example, myeloid-specific knockout in atherosclerosis. This is work, a PhD project of Yong Wang in my lab. And Yong has already shown this is an important, or this leads to reduction of the initial lesion formation and less or reduction on macrophages in the plaques, which is probably linked to a regulatory effect on macrophage proliferation, which is also following up in terms of the mechanisms. We are also interested in endothelial CB1 deficiency because we also have some interesting observation in a cell-specific, endothelial cell-specific deficiency model. And this is the project of Binny, a PhD student in my lab. There, for example, she's using a lot of imaging and using this imaging of an ex vivo perfusion of a carotid artery and then perfusion with fluorescent oxidized LDL. She was able to show that this you see much less oxidized LDL uptake in knockout arteries. So this is a three-dimensional reconstruction of a part of a carotid artery, which had been perfused with this oxidized LDL. So knockout of CB1 in endothelium has an impact on lipid uptake based on these findings we are following up. So so this is about the atherosclerosis part. So we have overall, we would say there is on the one hand side the pro-atherogenic phenotype of CB1 signaling and the atheroprotective function of CB2. So maybe even it might be interesting to have a combined agonist-antagonist strategy. Yeah, and in the next remaining few minutes, I would like to discuss the relevance of the cannabinoid signaling in myocardial infarction healing. We know already from quite some time that there is some effects of cardioprotection, and if we activate CB2, we and others have found that this limits ischemia reperfusion injury. So there's a protective signaling in cardiomyocytes, induction of pro-survival pathways. There's also an acute inhibition on neutrophils, less reactive oxygen, but we don't know so much what this receptor or cannabin signaling is doing in this more chronic inflammation and resolving phase and subsequent healing response, which is then potentially orchestrated also by these lipid mediators. And of course, given that the CB2 receptor is highly expressed on B cells, so far there was not much known or nothing known how this would affect the myocardial healing response. This is a project that a postdoc in my lab, Michael Hochmanns, was working on when he was still postdoc in my lab. And he's now back in Brussels and establishing his own group. And we are still collaborating on this project, what I would like to just highlight here now. So when we started this project, Michael came across this interesting paper here. So Cecil Benezek has shown here in this paper that in various fat depots throughout the mouse body, we have clusters which are full of immune cells. So there are a lot of B cells in these clusters. There are some T cells and a few myeloid cells. And she called these fat-associated lymphoid clusters, or short FALC. And what was really intriguing for us was here just this graph here, which compares the number of clusters per gram tissue. So tells you something about the density. And this red encircled here, the pericardial fat, has a huge density of clusters. This is one of the most enriched sites, I would say, in these lymphoid clusters. So we were wondering, well, these clusters, should they maybe have an important role in cardiac homeostasis, maybe immune surveillance, and what would be the role if we have an acute immune response to myocardial infarction? So this was the starting point here of this project. And to explain this a bit more in detail, maybe, because this is really a tiny piece of adipose tissue which we collect when we dissect the mice at the level of the apex. We have a tiny amount of pericardial adipose tissue, which then we can use entirely for staining and three-dimensional imaging by multiphoton microscopy, or we can do, for example, flow cytometry to look at the cellularity. And this maybe even gives you a better overview. If this is like really a standard simple H&E staining of an entire pericardial adipose tissue. The gross view here, and you see these dark spots. These are lymphoid clusters that you even already see in a wild-type mouse without any infarction, and this enlarged view of so-called FALC. And also really simply illustrates that the lymphocytes, the immune cells cluster, and you have a few cells in the surrounding. And when we use a flow cytometry, we can see that the leukocytes we find in these clusters, they are mainly B cells, There are less T cells and only few myeloid cells. Okay, so this is the schematic view. And then we went to look into the comparison between the CB2 knockout mice and the wild type mice. And we found already at baseline that we see much larger clusters. They are stained here with the CD19 antibody in red. And when we subject mice to myocardial infarction, these clusters become much bigger and even much bigger in the CB2 knockouts, suggesting an important role in this regulation of the B cell response in these lymphoid clusters. So to have a more quantitative view on this, this is flow cytometry. You see the comparison in terms of numbers of B cells per gram fat tissue. We see already at baseline this increased number in the CB2 knockout mice. And then we subject mice to myocardial infarction. We see an increase in expansion of B cells with the peak in the wild type mice at 3 days post-MI. The same is happening in the CB2 mice. Knockout mice, it's just that the response is much more pronounced. And then it drops afterwards. So this is also then, we went on mainly to look into this 3-day post-MI time point subsequently. So then if we look into the heart of the same mice, we asked, what is the relative contribution or recruitment or expansion of B cells in this fat tissue compared to the heart? This is somehow also what has been already shown, that there are very few B cells in the heart, and there's very few expansion also after myocardial infarction. Because B cells, yeah, they are involved in the adaptive immune response, so they are not really, they're not so much recruited to the infarct area to contribute to this immune response. Well, nevertheless, we see also in the CB2 knockout mice that there are more B cells, but yeah, when we try to calculate this, we would say there are much less B cells in the heart compared to the fat tissue. So we propose that this pericardial adipose tissue is a major site for this adaptive immune response and B cell activation after myocardial infarction. And yeah, among B cell subsets, there is an important subset which is able to produce GM-CSF. We started to look into this because we found that there is a huge upregulation of GM-CSF, especially in the CB2 knockout mice here. This is in the plasma. Of course, there are many cells that can produce GM-CSF and that are induced to produce GM-CSF after infarction, such as cardiac fibroblasts. So we went on to further stain specifically B cells in the pericardial tissue in combination with GM-CSF. So this tells us that there is an expansion specifically of B cells expressing GM-CSF in this tissue. And again, much more accelerated in CB2 knockouts, and further also did some additional control experiments, blocking experiments here in this case with a B cell depleting antibody, and this then reverses this expansion of the GM-CSF positive B cells. So what is known about these GM-CSF B cells? They have been reported by Phil Swierski in sepsis and pneumonia. He called them IRA B cells, which is innate response activator B cells. They amplify the inflammatory response to infection in these models. He also showed their importance in atherosclerosis, where they prime dendritic cells, they activate dendritic cells, induce Th1 polarization. It's overall in pro-inflammatory response. So, and the question is now, was for us to ask, are these GM-CSF positive B cells selectively activated in the pericardial adipose tissue, or could this also be more generally happening in lymphoid organs, and in particular in the spleen, because this is where these cells have been mainly described to be located. So what we did here now, the next step was to take wild type mice, we subjected them to MI, and then we looked into different compartments, the lymph node, the draining lymph node, pericardial adipose tissue, and the spleen. Then we quantified cell counts, and what you see is somehow as we would expect from the literature. We see a lot of these GM-CSF positive B cells in the spleen, but nothing is happening in response to myocardial infarction. So the number stays more or less the same. But when we look into the pericardial adipose tissue, these are the red bars here, we see that this is much increased in numbers selectively in this pericardial adipose tissue. Then we also went on and looked into other tissues. So this is really specific for pericardial adipose tissue. When we look, for example, in visceral adipose tissue, we find only very few B cells. So this is really seems to be a selective response in this fat tissue. So another question is certainly how are these cells then activated in the pericardial adipose tissue? Because there's obviously not a direct communication in terms of vascularization between the cardiomyocytes and the pericardial adipose tissue, so outside the pericardium. So we may speculate these are danger-associated molecules which are released systemically from the necrotic muscle after ischemia. There can be professional antigen-presenting cells that pick up antigens and then would travel to the pericardium. So typically dendritic cells. So we address the possibility that dendritic cells could migrate to the pericardial tissue. To do so, we did a tracking experiment here. In this case, we isolated the super wild type dendritic cells. So we isolated bone marrow cells, and then we differentiated dendritic cells and labeled them ex vivo with a cell tracker, and then we injected them at the point when we subjected mice to myocardial infarction, so directly into the myocardium, into this ischemic area. Then we asked, after 3 days, can we find these cells in pericardial tissue? And we did so, of course, otherwise I wouldn't tell you probably. So here, of course, this is the injection area in the myocardium, where you still see some of these injected dendritic cells, but we also found a lot of these cells in adipose tissue. And I should also say that in the spleen, we couldn't find any specific signal of these dendritic cells. So again, pointing to really a more specific capacity, so we can, at least from this experiment, learn that dendritic cells can migrate after infarction from this infarct area to the pericardial adipose tissue. So this might contribute to the activation of the immune response in these so-called FALCs. So yeah, to cut the long story short, what we found by different depletion experiments and yeah, cytokine profiles we've seen here is we found that there seems to be an activation via GM-CSF, which is a known inducer for cytokines and the cytokine pathway involving IL-23 and IL-17 production in T cells. And this is an important inducer of granulopoiesis. So IL-17 induces G-CSF in mice, and this leads to upregulation of granulopoiesis, GMPs, and then subsequently also neutrophil mobilization. So all this process was accelerated in the CB2 knockout mice. So and indicating that this pathway, the CB2 signaling, might be crucially involved in this induction of the granulopoiesis in this post-MI situation. So this was a combination of B cell depletion, for example, and cytokine blocking, one of the additional key experiments we did to link this really to the pericardial adipose tissue was to actually remove the pericardial adipose tissue during the MI surgery. And then when we asked afterwards, if we do so, mice are subjected to MI and we remove pericardial adipose tissue, what is then the effect on the granulopoiesis? So we looked into the bone marrow and we quantified by flow cytometry the neutrophil progenitors, and we found that there's a reduced number in response to MI when mice were depleted or were we had to remove the pericardial adipose tissue. And this was also reflected then after 3 days after myocardial infarction, when normally the bone marrow starts to has reproduced or produced de novo neutrophils, and then we had removed the pericardial adipose tissue. There we saw that we have also less cardiac neutrophils that are recruited to the infarct area, which is supporting actually this link between the pericardial adipose tissue inflammation, the cytokine systemic induction we see, which then can act on the bone marrow to induce granulopoiesis and then subsequently provide subsequent supply of neutrophils to further repair. So maybe at the end I would also like to share with you some of this imaging of the bone marrow we did, which tells us there's not only just the granulopoiesis, but the CB2 endocannabinoid signaling axis has a general impact on this on the stem cells. We see because you see the bone structure here. This is a multiphoton imaging where you have this where you can see the bone collagen structure. The lineage positive are the mature leukocytes we see in the bone marrow, and then the CD150 is a marker for hematopoietic stem cells in the bone marrow. So there are only few stem cells, of course, in the bone marrow, but we see that in CB2 knockout mice, we have much more of hematopoietic stem cells, which then also nicely illustrates this importance of this signaling axis in hematopoiesis. So, okay, so I would like also to add to this overview that not only in atherosclerosis, but also in this acute or post-acute inflammation response, there's an important crosstalk between innate and adaptive immune response. And we think that the pericardial adipose tissue is an important location where this is happening, and that this endocannabinoid signaling axis has also important implications in the bone marrow hematopoiesis. So, and yeah, then to come to this probably most important slide, as always, I would like to acknowledge my team with current lab members, postdocs. I've shown you data where mainly from Raquel on atherosclerosis. We have, of course, other projects ongoing on GPR55 where Sarah is involved. We have several PhD students. I pointed out some of them. And I would like to, of course, acknowledge specifically Michael Horkmans, former postdoc in the lab, who is involved in the pericardial adipose tissue project. Of course, we have local collaborators at the Institute, and in Brussels, we have also collaboration with Finnish people for carotid artery plaque data, and for endocannabinoid measurement in Germany, the University of Mainz, and also from Italy, we had a nice collaboration about epicardial adipose tissue, where we actually also looked into these B cell clusters. And of course, the funding bodies. And yeah, I think I'm going to stop here and happy to answer questions if there's any.
Okay, thank you very much, Sabine. Can you stop sharing your screen? Then we can see everyone again. Yeah. Okay, it was a beautiful presentation. Are there any questions? I see Mabruka. She's raising her hand. Yeah.
Hi.
Mabruka, you go first.
Yeah. It's very, very great presentation. So I might have missed this at the beginning of your talk, but are both SP1 and SP2 being activated by the same ligand? And second question is, seems like CB2 signals through MAGL inhibit CB1, but how that is happening? Is it direct or there's some mediators are involved in the middle between the two? Thank you.
Okay, so yeah, this is, so as to the endogenous ligands, they have receptor binding efficacy based on, pharmacological assays like receptor ligand binding to both receptors. Then, of course, there are specific compounds that would activate, synthetic compounds that can activate specifically one receptor. So I would say endogenously, it's due to probably the presence of the receptor and other ligands. So it's, if we selectively target some pathways, for example, also the other pathways with fatty acid amide hydrolase, we see effects that we would partially link to the CB1 receptor, at least, for example, on smooth muscle cells, we could prove that there is some effect of the other endocannabinoid that is promoting a more inflammatory response, neointima formation. This is CB1 dependent. So might be maybe also on this local production, which of the ligands is produced, availability of the receptor. Yeah, this is, and then there could be also functional desensitization of one of the receptors. This was your second question, how we explain this effect on the CB1.
Yeah, through MAGL, it seems like how that is happening. Is it like at cellular level, or is there a mediator that's involved between the two and it goes back and where it regulates the second receptor expression?
You mean this desensitization process?
Yeah.
I think probably this is, well, this is mainly studied in vitro systems with reporter cells or specific knockdown, for example, of phosphorylation sites and so on. So there's a lot of, this has been shown in CB1 cells. This is always in this specific system. Whether this would be then true, for example, in inflammatory cells or in endothelial cells, can only speculate. So far, we would like to translate this also to a more relevant cell type, whether the signaling is happening in this way. But this is another in vitro project which we are working on. Yeah.
Very beautiful work. Thank you.
Okay. Jesse, I saw you waving.
Yeah, Sabin, nice talk. I was really surprised by the migratory pattern you showed for the transfer model with the, I guess, dendritic cells moving away from the injury site toward the pericardial cavity. And that seems to be kind of opposite of what Paul Kubes showed in that immunity paper in 2019, where he showed or suggested that the pericardial cavity macrophages were infiltrating into the lesions. I was wondering if you guys had any mechanism of how these cells might be moving and whether it happens without the injection, because it could be possible that the injection itself is, the transfer is causing some of it.
Yeah, so this Paul Kubes paper you're referring to, he's looking in the pericardial cavity fluid. And then he has some models where he is ligating without opening the pericardial cavity, comparing this. So this assumption is probably that in this fluid cavity, you can have some cells. Of course, this is somehow the problem of the model. This is a surgery model. So we do not yet have I don't know, would be nice to move to a less invasive model, which, for example, this recent work from, I think Florian Leuchner just published a model where they could, without having this opening, the thoracotomy and the pericardium open. So there might be some effect. So this is a different location where they looked into.
Yeah, I was thinking of whether perhaps you had done GPCR blockade potentially with a drug to show
global, no. What we've done in terms of the dendritic cells, we have also used, for example, CCR7 knockouts. So we see that we have a high expression of CCR7 in the pericardial adipose tissue dendritic cells. These are the most mature dendritic cells we found. And if we have a CCR7 chemokine receptor knockout, we find less infiltrating into the pericardial adipose tissue compared to the heart. So in the heart, we didn't see changes in the CCR7 chemokine receptor knockout compared to wild type in terms of dendritic cell numbers. So this may argue, but of course we haven't directly like in vivo tracking or something like this. And this is something more work to be done.
Okay, Marit, you are next.
Thank you, Sabine. Very, very nice presentation. So I was wondering, you showed some data on the CB1, so the ApoE knockout, CB1 knockout, if I remember correctly, where you saw effects on the CXCL12 and the SCF1. And then later you showed this nice staining that you do on stem cells in the bone marrow. And I was wondering if you had already also done that in that particular model, and if you also are looking into stem cell mobilization, whether that is effective.
With the CB1 knockouts, yeah. This is George is working on this, and we have we obtained a reporter mouse where we try to image. This is in the work in process, establishing process, I would say. Well, just if we would compare the knockout to the wild type, so this we can of course just do and look in quantitative just by staining HSCs, for example, or the overall structure of the niche. This is something to be done, of course, but we would also it's not clear for us. So there might be a contribution which is just due to this innervation of the bone marrow, which then affects stromal cells in the niche that makes that there's, for example, less CXCL12. Or is there an intrinsic role of hematopoietic cells or progenitors as well? So
thank you. Very interesting work. Yeah, thank you.
Okay, Lynn is next.
Yes.
Sorry.
This is actually Klaus. Nice talk. So my question is related to Jesse's. In this experimental setup with the dendritic cells, how do you know that the dendritic cells in the adipose tissue came from the heart? And how do you know they didn't see directly after you injected the precursors?
Yeah. Well, you mean, so I mean, of course, so the injection, so this transfer experiment, this was, I would say, more this direct, really in the really, we went with the needle into the infarct area, right? So it was not a systemic or IV or something injection. So this was really locally applied in the infarct area. I don't know if this might address. This is, I would say, a hint. It's not a perfect proof, but this, yeah.
And do you have a time course? I mean, how long does it take for them to migrate? Do you know?
I don't remember. I would say we probably also went 3 days after this infarct area, but for sure we didn't do a kinetics, a time course to look into this.
Okay, thank you.
Ada, you have a question? Yeah.
I was wondering about the difference between the pericardial adipose tissue and the perivascular adipose tissue, and whether maybe in the athero model, the perivascular is more important.
Sure, yeah. So, well, also in humans, what would you? So in mice, we can study. What can we study is the thoracic probably part and the lower abdominal part of adipose tissue. In humans, we have epicardial fat, which is around the coronary arteries. Mice barely don't develop any epicardial tissue. So certainly there's a local crosstalk with the plaque. So there's concepts where people say this inflammation from the plaque in the coronary vessel can affect the adipocytes on the outside. So these adipocytes are more, I think, more immature. There is work from in Oxford. People are working on this, for example. Certainly in terms of inflammation. So if we think of these clusters here, we find them also in the epicardial tissue around these coronary vessels. If you think of this chronic atherogenic process in the mouse model, there is also work which is also here at our institute. A group studying in aging. You have a development. Think in this perirenal area, so in the lower thoracic or abdominal part, the development of these so-called ATLOs. So, well, these are more located in the adventitia, but there is certainly also a crosstalk. But this is more directly related or connected, I would say, to the vessel, where you have also immune cell clusters. So the intriguing part for us is also, or the difficult part is to understand for us that this pericardial tissue, which is outside the pericardium, so there is no direct microcirculation between the myocardium and this adipose tissue. So how is there the communication? Or could still there be a sort of of a gradient that could be transmitted between the myocardium and adipose tissue and the other way around? But we would not expect that these tissues, the immune cells, would directly go through the pericardium to enter the heart. So it would go via maybe circulation or lymphatics.
Okay, thank you. Sabine, I think the last question is CB2, is that expressed on quiescent HSPCs, or is it just an indirect effect that you don't see it, or is it your receptor on a marker on quiescent HSPCs?
Trying to remember if we have looked into, we've sorted cells recently, and we are looking into different genes. We actually do this mostly because we have a problem with specific antibodies, but I can't tell you what was the readout of this. Maybe Raquel would know, but maybe I can answer you later on.
Okay. Are there any more questions for Sabine? Then, if not, I would like to thank all of you for logging in, and I hope you will be with us next week, same time, same place. I wish you all a happy rest of the day, nice evening, and a nice weekend. Okay, see you next week. Bye.
Bye.